Sara.
Cool. So my first question is, do you want me to use the microphone, or would you rather I not use the microphone? This is new
For
data.
the filming, it's better.
Okay, so I've got it comes up from the tech guy. It says, yes, please use the microphone. All right. I'm not like overpowering the room with my, bombing voice, so that's fantastic. All right, so thank you, Elena, for the introduction. Like I said, like she said, I'm Sara Goodwin. I've been with Cold Spring Harbor for a few years now. I've been working with the Oxford Nanopore device for about 2 years. We've had the opportunity to do a couple of really interesting experiments with it. My kind of role in the lab is to evaluate new technologies as they come out. So, we have Oxford Nanopore, Packed Bioillumina, all these different things that you'll be learning about either already or later on through the length of this course here. So we'll be spending the next few hours specifically on this kind of very new third, fourth generation sequencing platform that has been kind of setting the sequencing field abuzz lately. So we're going to start at the very beginning. I think it's a very good place to start. So the cell. Now this is really very much the beginning. I think, as you all know, the cell is an aqueous bag surrounded by a phospholipid bilayer in which we have head groups and tail groups. And those tail groups and head groups prevent the movement of water in and out of the cell. They're going to keep a seal around it. Now, problematically, that means that items like sodium and ions that are important for the cell, they can't get through. We don't have easy diffusion of any of these charged particles through the cell. This is going to include anions, cations, larger molecules, larger nutrients, different things the cell needs to carry out its day-to-day activities. Fortunately, there are ion channels. Now, these are protein structures embedded in the phospholipid membrane. This shouldn't be new material for you guys, but if you have questions, don't hesitate to ask. And these are going to allow a permeability of these charged particles through the phospholipid bilayer, which generally does not allow for the diffusion of particles. Some of these channels are just open and closing at particular rates, some on, some off, allowing for passive movement of the molecules through it. Some are going to be ligand-gated channels. These are going to be requiring some other factor to bind to them. Sodium-potassium pump, for example, requires certain binding of the ions into the channel to open and close. Others require acetylcholine to open and close. And that, again, allows for the diffusion of important ions through the membrane. You have voltage-gated channels. So those are fairly interesting because they build up of a particular membrane potential on one side or the other of the cell membrane. This is what drives all your synaptic processes. So it's a very important feature present in many channels. Now this is important for nanopore sequencing because ultimately we're relying on voltage and membrane potential for driving the sequencing through that nanopore. Now, ultimately, a lot of people did a lot of research on these channels. A lot of information was discovered about these channels through voltage and patch clamps. This is going to be using the oocyte frequently of this nice little froggy up there. I believe he's called Xenopus. And what that does is they apply a voltage current to the cell through either stabbing something into the cell or actually putting a specialized pipette onto the cell. In fact, this guy, I think I have a laser pointer here, yeah. This is going to be your voltage clamp. That's going to be your patch clamp type structure. And what this allows us to do, what allows researchers to do, is to measure the change in potential while holding voltage constant and seeing what happens to the membrane potential as those channels open and close, as they allow the diffusion of ions through or out of, into or out of the cell. Now this was very important because it allowed for the discovery, greater information about what actually happens when ions are present inside or outside the cell. Now, furthermore, the current across the cell can be measured. So what we can see is we can see a diffusion. Here's we have a pore open, and then we see a blockade. Open, so there's more current passing here. Now it's not passing. More passing, not passing. So the current, through research in electrobiology, we can measure, it's very, very small, but we can measure it across channels. So all this work, all this building up of information about channels, patch clamping, voltage clamping, understanding how these things work, has allowed for the discovery of some very interesting proteins that are used as pores in different organisms. Now, one is alpha-hemolysin. This is a toxin from Staphylococcus aureus. It's a heptameric beta-barrel porin. It is 5 angstroms at its narrowest, which is actually 1 to 2 nanometers wide. That's actually pretty darn big for a channel. Now it has 2 modes of action depending on its concentration, but fundamentally when Staphylococcus aureus wishes to kill something, it secretes some of this toxin which self-assembles into the membrane of its target cells, allowing for the movement of monovalent ions and calcium, ultimately resulting in apoptotic processes. The cell is full of ions, it loses its ions, it lyses, it pops, it's dead. Very happy Staphylococcus aureus. Now, interestingly, we can pass DNA through this pore. There's been a fair amount of research on this particular protein, at passing proteins, DNAs, other oligomeric material through it. That's because this pore is capable of remaining open for very, very long periods of time and accommodating relatively large molecules through its pore. Now, another protein of interest is the MSPA, which is a porin derived from Mycobacterium schmegmatis. Mycobacterium schmegmatis A, porin A. This is an octameric beta-barrel structure, which 1.2 nanometers at its narrowest. So like alpha-hemolysin, it remains open for a very, very long period of time, and it allows for the movement of very large molecules through it. Now, one major difference is this length here. This is the neck portion of the protein. It's much shorter than that present on the alpha-hemolysin, so that's going to change how the material is measured, how the voltage is blocked. And we'll get to what I mean by that shortly, as the DNA or whatever material translocates through the pore. Now, like the alpha-hemolysin, it's a negatively charged internal radius, which is great for DNA. We wouldn't want a positively charged internal core, because then our DNA wouldn't translocate through it. And this is important for the organism itself, because this allows for the diffusion of hydrophilic nutrients into the cell. So when the cell needs things, relatively large molecules in the world of molecules, this allows for the diffusion of those into the cell. All right, now, not to be outdone, this is obviously not a protein. People doing research into nanopores, understanding channels, wanting to know how large particles travel through proteins. The chemists out there says, no, we can do this too. And they have been working on graphene nanopores. And the graphene nanopore is fundamentally different than a protein nanopore, because we actually have this carbon graphene structure across a voltage current with tin, nitrogen. I don't know everything that's going on. I'm not a chemist. But these are actually using lasers or other various micro-machining techniques driven into these very, very thin graphene surfaces. Now, because of the conductivity of the structure of the membrane through the graphene and the tin and silicon and whatnot, we can see as DNA translocates through it, let me see if I can explain some of these figures here, similar changes in current. So you saw the current changes, the natural pore as it's opening and closing. Similar thing can occur as DNA translocates. We functionally block the graphene channel as a DNA or other molecule passes through it. Now, there's different speeds where things pass through. That's what this guy over here is. These counts. That's sampling. These aren't constant things that are carried on. We don't have a continuous value of measuring the current. That's actually a sample. So it's measure, 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 measure. Anyone familiar with computer science recognizes what a sample rate is. So that's what these counts are. And it says it's translocating. Here's a better resolution. DNA is passing through, passing through, passing through. One of the problems with graphene pores, unlike what's present in those alpha-hemolysin or MSPA pores, is that it's no biological driving force for why the DNA translocates through the pore. There's no natural interaction between the amino acid residues and the DNA structure. There's no charge that's intrinsic to the structure itself. These are things that are engineered into it by people, as opposed to kind of the natural engineering that proteins go over, which tend, at least right now, to be better than what we can create de novo on our own. So DNA structure has a real major impact on these graphene nanopores. So if the DNA is folded or bent or circular, it changes the way the DNA behaves as it travels through the pore. With the protein structures, we can actually incorporate additional material, which again we'll touch on in just a few minutes, to help facilitate the DNA as it translocates through those pores.
Sarah, is the other challenge of graphene just the ability to machine uniform pores as alpha hemolysin, which is always going to make itself look exactly the same every time.
Absolutely. And I didn't actually include the figures, but there's from this manuscript here, which I'm happy to share any of the manuscripts I show during this session, there's a number of pores that do not show the consistent behavior. Now, without consistency, while you can measure DNA as a translocase for the pore, the voltage is no longer characteristic. So one pore, it's characteristic, but your next pore over, and you got to measure it individually as well, which really tends to limit the functionality of these.
And maybe just to finish this, so does everybody know what graphene is? It's single atom carbon. And the reason for sort of pursuing this, I think, tell me if I'm wrong, is that from a manufacturing standpoint, right, making a chip with alpha hemolysin pores is kind of a, as the Brits say, bespoke. Does that sound like right, Jonathan? It's sort of like I'm never sure if I'm saying that word right, but it's kind of like a custom bespoke. See, I wasn't saying it right. Bespoke. So it's sort of like a custom tailored thing, whereas this potentially could scale. You could potentially get lots and lots of pores in the same sheet, so you have scalability and also manufacturing ease. And I would guess maybe even a shipping stability.
Shipping stability is definitely something that this would have far and above over the protein pores, which those of us who've worked with the nanopore have suffered through very much. These ones, they don't need to be hot, they don't need to be cold, they can ship in ambient temperatures. All right, so I've talked about voltages, I've talked about blockades, I've talked about this, but I haven't explained it. What is actually going on with these nanopores that are embedded into a membrane? So as I mentioned, there's a membrane potential, the change of ions as they travel across from one side to the other. That's this little cloud here, that's what this represents. And there's a current, so long as the concentration of ions remains fairly constant on one side or the other, we can measure the current as it passes through on a local, very small level. As we stick things, small things in this case, into the pore, the ion flow through that pore is decreased. That represents a characteristic shift in the current as the ions are traveling through the pore. We can stick larger things in those pores, which can create a larger blockade of the current as it passes through. So that's characteristic of the molecule that's actually sitting inside this pore. Furthermore, the length of time the particle remains in the pore is also relevant for that. So if we have something that stays in the pore for a long time, the length of that blockade is also increased. And these are two features that can be measured to help identify what the molecule is actually sitting inside that pore. That's a characteristic feature. So some of the early attempts of this, now this is not the very first paper kind of touching on nanopore. There's actually a Church paper from 1995, but I actually like this one. This is a kind of a follow-up to that original research, but this is actually starting with those proteins, the very earliest attempts at trying to see what happens when we stick DNA inside one of these pores. And this is the Deemer group. This is from 1996. So we're looking for the voltage blockade. That's, like I mentioned before, a particle within the nanopore causing a change in current as the protein moves through. So in this case, they used poly-U. And on the cis side, that's where the DNA is being fed into. They see as a current here, they're applying 120 millivolts. Then they apply this poly-U to the cis side of the membrane. So they have a membrane, they have proteins that are self-organized into it, and it's separated from the trans side. So there's no flow from cis to trans except through the pore. That's important to take note. As the poly-U is applied, we start seeing these characteristic shifts. Okay, so a current of initially 120 picoamps is measured, and then we see a change in that as the DNA goes into the pore. Now here's a zoom in of that particular change. There's actually a shift of 50 picoamps as those pieces of DNA enter into that alpha hemolysin pore in this case. Now they started trying to understand the characteristics, the relationships between as DNA as it translocates to the pore with different characteristics of DNA itself. So unsurprisingly, the larger the piece of DNA, the longer the dwell period was. So the DNA remained in the pore for longer periods of time, the longer it was. Not all that surprising in and of itself. And these different lines here represent different populations of DNA they isolated from their process. They're all poly-U. There's some structural differences between them though. Over here, we see that there's, but actually I believe I have animations for this. So fragment length is also inversely proportional to dwell time. So that is voltage, I apologize. So inversely proportional to voltage, where the longer the voltage, more voltage is applied, the faster that DNA is translocating through the pore. So it's in there less time, the more voltage is applied. Anyone who's run an agarose gel knows if you crank that voltage up on the agarose gel, that DNA zips right through. Of course, you might melt your gel, but details aside, the voltage makes DNA move faster. And finally, fragment length is proportional. I don't know why I have the three things all in the same spot here. I apologize, that makes no sense. So the blockades per second is proportional to dwell time. So what they did here, to explain what this experiment was, they took a long 10,000 base pair poly-U fragment. This is produced synthetically, by the way. And then they added ribonuclease A to this. So the very, very large fragment does not translocate through the pore for diffusion reasons and structural reasons. It doesn't do anything. And that's at time point 0. Then they add ribonuclease A, which starts breaking up that molecule. As the molecule gets broken up into smaller and smaller pieces, we start seeing an increase in the number of blockades. Yes, one of these guys here. That's because as we have smaller pieces and there are more pieces, the number of pieces capable of translocating to the pore increases. So basically this implies that the molar concentration and the particle size is relevant to the behavior as the DNA translocates to the pore. As the DNA is depleted and turns into smaller and smaller fragments, eventually down to just a few base pairs, you no longer have such a significant blockade event, and it goes below the limit of detection for the pore. This guy here is poly A. This was not a substrate for the ribonuclease A molecule they used. It never went into the pore. The number of blockades never went up because it was never broken up into smaller pieces. All right. That aside, that really doesn't tell us anything about the structure of the DNA. Yes, we can stick some DNA in there. Gee, it makes the voltage change. Not all that meaningful. The authors of the previous paper did suggest it could be used for other applications in addition to DNA sequencing, for example, measuring hydrolysis, which is what we saw in that last figure. But moving on, we wanted to see what, and excuse me just 1 second, my computer charge is running low. I don't want to lose my presentation. Much better. Okay, so now I want to see, can we distinguish between nucleotides as they pass the pore? We can see from the previous paper that, yes, we can see that there's something in the pore, but not what it actually is. So what this group did was they created these kind of hanging polynucleotides. They took a biotin, they stuck these poly Cs with a string of A's in the middle, and they stuck it in the pore. That's allowed them to see the position of the A's in different locations in this alpha hemolysin pore. And what do you know? But there is a difference in the characteristic shift in voltage when the poly C is in there versus when the poly A is in there. However, it's not that different. So this is the residual current, this is the percent change in difference. What they can do is you can have a mutant organism. Now this is changing some of those internal residues inside the barrel of the nanopore from a charged amino acid to a polar amino acid. This is going to allow facilitate DNA translocating through more easily. And you can now see a much more significant shift there. The differences between poly A and poly C not only are flipped from wild type, but they're also more easy to distinguish between them. So changing some things on the molecular level of the protein can also facilitate the ability of these proteins to distinguish between nucleotides. They are also able to see that different portions of the pore here have different ability to detect the polynucleotides. In fact, the most sensitive region is right here. Okay, now what this figure is showing, this is the percent change in voltage relative to just poly C. So as you put poly C in there, we have one change. As you stick the poly A's in there, that's different changes there. That's where we see the back and forths. We have an increase, and we actually have more voltage going through. That's where we see the back and forth lines there. Those are poly A's, so they're looking at 5 different residues. They also do the same thing with single nucleotides. This is the same thing. I don't think you can quite make out what's going on here, but this is poly C's with A's at different positions within the chain. Again, hung down to that pore, and at the same, or roughly the same position within the pore here, we see a most significant deflection of the current, the most characteristic deflection of the current. So not only do we have polynucleotide resolution, we have mononucleotide resolution within these pores. Furthermore, and their final experiment in this particular suite of experiments was instead of putting A at all these different positions, they picked the best position right here, and they put an A, T, G, or C in that spot. And each individual base has its own characteristic voltage shift histogram. That gives you single nucleotide resolution. Now that's the first sequencing. We can know the sequence of the DNA as it goes in the pore. Now fast forward to 2012. Okay, 2012, Oxford Nanopore comes to AGBT. We start seeing headlines, headlines, headlines. Wow, wow. Everyone says this is the most awesome thing in the whole wide world. Going to demolish Illumina, demolish everyone else. Yay. And then we waited 2 years. So 2 years later, 2014, that's when the MAP program eventually actually came to play. The MAP is the MinION Early Access Program. A number of labs across the world gained access to the Oxford Nanopore MinION sequencing device. This allowed researchers to evaluate and experiment with this new nanopore-based sequencer, which was announced 2 years ago, but then no one saw anything for a while. Certainly, MAP is still open. Anyone after this talk, if you are totally enthused about the Oxford Nanopore device, you can most certainly sign up for MinION and get your own nanopore and do your own sequencing right at your lab. Yes?
Can you go back 2 slides? Just one question.
Sure.
On the bottom right. So this percentage is that, what this is saying is that when the C is passing through this modified alpha hemolysis thing, the current is reduced to a 35 is lower
yes.
when a G is passing through.
Correct.
So it's still not a huge difference between CT, AG. It's not a gigantic difference, but any of them, all 4 bases reduce the current to about 35%, but there's slight differences between.
And it's actually a very, very good point. We'll actually kind of touch on that, how these small differences are actually a big problem for these sorts of devices. It's caused some no end of heartache to the people who are trying to build algorithms for solving this. And we'll definitely touch on that point in just a little bit here. All right, so again, what I was saying, if you want to join MAP, by all means, get your hands on a Mark 1 MinION and start sequencing at home if you want. I've seen pictures of that next to the coffee table. It's kind of funny. All right, the MinION device, what is it? All right, this is a small device about the size of a Snickers bar, perhaps. Within the device, we have a variety of different pieces. We have the electronic systems. We have an internal cooling device. The shell, the flow cell itself fits right on in there. It's really very, very small. And in fact, I'll have an opportunity, in fact, if y'all want, to kind of see what one of these instruments look like. This is actually a Mark 1 MinION. But I'll hand this around. You guys can take a look at it. There we go. Alrighty, now of course what you're seeing now is the Mark 1 MinION. That's this guy here. This was the early access MinION. These guys have been retired now. They've really improved the electronics and the cooling system, for example, in these devices, and the way the flow cell actually nests into it. There will be a Mark 2 sometime in the future, which is ill-defined, but this is where we are now. That's what's being passed around the room. And this operates through a USB device into a laptop. Now the computing requirements for this guy really aren't that bad. We're not expecting a supercomputing system to be able to run these guys. The most touchy guy is this one here. This is the solid-state hard drive, which are getting more and more common now. It's not that unusual to have these things. And it says greater than 120 gigabases. That's the minimum required. If you want to actually do sequencing on this, you need a lot more. I would say you probably need close to a terabyte at least. The file structure that's generated off the FAST5, the MinION data structures, are really quite memory space intensive. I can generate many gigs of data off of a single run. So if you want to store anything on the computer for any length of time and do some internal base calling or analysis, you really need that extra space. Otherwise, everything is very standard. Currently, there is not Mac support. I know a lot of you are Mac fans. It's getting there eventually. I believe there are some hacks that exist for making it work on Mac, but right now it's a PC-only instrument. All right, so within that MinION device, we have a flow cell. That's me holding a flow cell. It's very, very small. It fits in the palm of your hand. Within that flow cell is a synthetic membrane here. So it's not a phospholipid bilayer. It's something different. They won't say exactly what it is, but they're very clear it's not a lipid bilayer. Okay, that's fine. Within that, there are 512 channels. This is the current Mark I structure. Within those 512 channels, there are 2,048 individual proteins in groups of 4, okay? So 4 in each channel. The proteins are, as I said, self-assembled into the synthetic membrane, but you can only collect data from 4 proteins at a time. So what's going on here is in each channel, there are 4 proteins, and then you can measure information from one of those proteins, and then you can switch to a different protein, but you can't measure from all 4 at once. There's a couple of things. You can switch it manually, or you can switch it using scripts at special times, or you can allow the software to switch it at 24 hours. The scripts are not from Oxford, though. The scripts are from MAP community members. All right, so within each of those dots, we have the little protein pore on there. Now, there are actually 2 proteins involved. I'll kind of touch on what those are shortly. When the DNA translocates through the pore, we measure the change in current. We have DNA sequencing. Proteins. Oxford Nanopore uses alpha hemolysin A. However, they did investigate many hundreds of proteins for use in this system, including MSPA, other proteins I don't necessarily know, mutant versions of these proteins, and this is what they have settled on for their current approach. The different proteins change how fast the DNA translocates through it, what type of structure, how it passes through, what the processivity is, how many bases can pass through it. The the alpha hemolysin is what they decided was the best choice for their initial rollout of this structure. There also are other proteins involved in library preparation, which presumably they also included in their testing for performance of the proteins in this application. So fundamentally, the Oxford Nanopore is an alpha hemolysin non-destructive DNA sequencer. The cis voltage drives the DNA through the pore. The motor protein, that's this guy here, facilitates DNA unwinding and facilitates translocation speed. I don't know what this protein is. It's a mutant something, but it does control the speed. Maybe it's a helicase. It's probably a helicase of some sort. But that was what's going on. That's one of the proteins that's involved in the translocation. And the ions through the pore change the membrane potential locally, and you measure current. So how do we do things? What do we do? This is the workflow for how to do MinION sequencing. You start with DNA or cDNA. There actually is an approach that uses DNA RNA hybrids. In my hands, it hasn't worked very well. Maybe you guys can make it work better. But DNA or cDNA, sample preparation, about 2-hour library preparation time. Takes about a minute to add to the MinION, and then you have real-time sequencing data. So there's a lot of feedback. You actually get results right away. And depending on what application you're doing, you can actually even start analysis as the data comes off the instrument. Run times range from just a few hours to about 48 hours currently. There's a really fast process to get the data off the machine. So we're going to go through what the library preparation is real quick like. Start with your DNA, and you're going to fragment that. Now I use Kavaris G tubes to fragment. Those devices allow me to fragment anywhere from a few thousand to 10,000 plus bases. You can use unfragmented DNA. Some people have. One of the problems of using DNA much in excess of 10,000 is it becomes fragile, and you have a lot more difficult time properly forming the library and delivering it to the pores themselves. We have a pre-CR or FFP step, which is optional. This takes about 30 minutes to run through. But this is going to repair any damage present on the DNA, which is going to inhibit the processivity of the DNA through the nanopore. Pre-Sara is what they've been using. It's what I still am currently using. They've recently moved to recommending this FFPE repair step. I haven't tried it out, so I can't speak to how well that is, but I'm hearing anecdotally that it's quite good. We now have a combined end repair A-tail step, which takes about 20 minutes. We have a ligation and barcoding step, and this is for non-amplified pieces. You can do amplification steps as well, but you can add barcodes onto your sample. These are going to be ligating your adapters onto it, here and here, and I'll touch on the adapters momentarily. And finally, we're going to select those properly formed library particles and load it onto the flow cell. That's about a 2-hour workflow if you're really focused on what you're doing. Now, it takes me probably close to 3 hours to do it, just because I am not gotta do it right now. I don't like making mistakes, but 2 hours, that's doing good. Now, this step here is one of the most touchy steps in the process. The reason is that there's no way currently that they direct the definite formation of the properly formed library. The properly formed library is this guy right here. We have a leader adapter, we have a hairpin adapter. This facilitates proper bidirectional sequencing. However, because it's a ligation with all the adapters mixed together, we have this hairpin, and this leader structure that's also present. The other important thing to note that makes this a more touchy step is there's some proteins pre-ligated in different positions, depending on which chemistry you're using, onto those adapters. So this is a fairly touchy, you have to use some of their fairly specific low protein binding tubes or special elution buffer. Treat it very nicely to make sure you get as much of those properly formed libraries as possible. Now, the important reason why they have these two different adapters is because this allows for bidirectional sequencing. What that means is the leader adapter is the piece of DNA that actually interacts with the initial protein and interacts with the pore. This helps direct the DNA into the pore of the protein. Then we sequence across the template strand until we reach the hairpin. Now the hairpin, if anyone is familiar with PacBio, allows sequencing all the way through. It's a continuous piece of DNA that allows for unwinding and then sequencing in the complement direction. So we actually get 2 reads off one molecule of DNA, both forward and reverse. Now, this guy here, the leader structure, this is still sequenceable. This will still result in data. However, we don't have that hairpin here. So we can't actually go and do the other directional sequencing. We just get the 1D reads. This guy, lacking the leader adapter structure, isn't directed into the pore, and we don't actually get any sequencing from it. Now, to help improve the number of those properly formed molecules, what they've added, this is, I guess maybe a year ago now, yeah, definitely more than a year ago, this his tag to that hairpin structure. This allows for his tag purification. We're just pulling down those molecules that have that tag on it. So this guy is gone entirely. Even though he's sequenceable, we're not all that interested in him because he doesn't have the bidirectional sequencing. This one is still there, but we can't sequence it, so we don't see it. And finally, this one. So in theory, all of your molecules in your final library should be properly formed and be able to undergo bidirectional sequencing. In practice, however, you still get monodirectional sequencing. You'll get discontinuous sequencing. It won't go all the way through the DNA molecule. You get computational problems where maybe it did do bidirectional, but it couldn't find the hairpin. It's possible some of these guys are coming through the mix. There's contamination, a number of reasons, but it is better than not using the his tag. All right, one additional feature of this is a tether. Now the tether is a complementary binding DNA sequence that binds to that leader sequence. It's embedded partially into the membrane, or a complementary sequence is embedded into the membrane with a cholesterol linker. This brings the functional concentration at the surface of the nanopore much, much higher than through simple diffusion. Through simple diffusion, we have these guys all floating around, and eventually one of them will interact with the pore and be sequenced. By introducing the tether in there, we're bringing more and more of those molecules and keeping them right there at the surface of the flow cell, increasing the probability that these molecules will interact with the pore. Oxford says, and I changed this slide from Oxford, they said 10 nanograms of DNA. In my experience, for large fragments, 10,000 base pair fragments, it's closer to 50 nanograms of DNA is a good amount to put on there. That's actually quite a lot. You start with 1.5 micrograms for library preparation, which means that you have to really have a lot of starting material. You have to be able to get a lot of DNA from your organisms to do this prep. There are some low input prep kits. There's a newly released one that came out in the last few weeks. I haven't tried. Perhaps this totally revolutionizes low input DNA, but right now my preference is to stick with 1.5 micrograms. Supposedly, you can trade concentration for runtime. The lower concentrated it is, in theory, eventually the DNA will get to the pore. Of course, the flow cells do have a limited life. You can't run them for 96 hours and expect them to perform as well at hour 96 versus hour 1. All right, data. What's actually going on? We've talked about DNA, we've talked about pore, we've talked about things going into the flow cell. Here's what's going on. We have a current. There's squiggles. We have squiggle space, okay? That's really what they call it, squiggle space. These are the changes in current as the DNA moves through it. However, all of those pieces are important features that represent DNA as it's translocating to the pore. What they do, now this blue area is expanded here, is they find what the structure is of each of those peaks, and then they can actually transmit it to the signal, the information to the base caller. Now remember what I said about sampling. So each of these points here is a sample, a value, dun, 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 dun. It's got a value, it's got a standard deviation over time. These are all put together to figure out what the signal is. Now, we're going to talk about how they figure that out. You saw squiggles, you saw lines, not necessarily useful. You mentioned that single bases are very difficult to resolve, and you're absolutely right. Oxford relies on a 5-mer base calling, so there are 1,064 different states that can go through. So you got AAAAA, you got AAAAG. Those are 5-mers. Mark 1 has now moved to 6-mer base calling. They use a hidden Markov model to identify what those bases are. So they take all those samples, they figure what they are, and they say, the average of all these samples together, what does that look like? To help explain what a hidden Markov model is, I'm going to go over a toy here, the loaded dice toy. A hidden Markov model is some situation where you just have the values, but you don't know the state that's producing those values. So let's say I have a loaded dice and I have a regular dice. And the guy rolling the dice is in a room, locked in a room, and he puts his number into the computer, and all you get is the numbers from the computer. Now he rolls the dice, he rolls the dice, he rolls the dice, and at some point in time, he switches to a loaded dice. Now, you don't know when that happened. And he rolls it and rolls it and rolls it, and then he switches back. How do you know when he switched from the regular dice to the loaded dice? All we have is these numbers here. Well, we can do is we can plot them. We go through all the rolls, dun, 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 dun. Values 1 through 6. Now you can probably see some changes here. They're not perfect. You can kind of guess, because this is the toy, where the loaded dice is occurring. But what we can do is we can take a window average. So we can take all those samples. This is like the sampling. Each number is a sample of the state. And we can average them together and create a path through the data using a sliding window where we average every 5 value together and plot. This is a smoothing function. So this brings outliers in to get an idea of what the local average value is during the course of the run, or in this case, the course of the die rolling experiment. What we end up seeing is we're seeing these transition periods here and here, here and here. This is where my random number generator, I changed what the parameters were and started only generating loaded values, okay? Now I know that this was the unloaded state, the loaded state, back to unloaded, loaded, unloaded. Now, there's 1,064 different states of the 5-mer, so this is a much more complicated problem than what I've described here, because there's only 2 states. But it actually does work. There's one other overlaying feature onto this hidden Markov problem that is helping them differentiate what the state is. And this is the verbiti algorithm that they're using. Because DNA is, by its sheer nature, ordered. So one 5-mer, or 6-mer, as it passes through the pore, is related to the next 5-mer as it passes through the pore. So if I have, in this case, TAGG, the next 5-mer must be something close to it. For example, AGG something. We don't know what that last base is, so any one of these 4 is a possibility, but there's only 4 possible new states the next signal can come from. All right? Now, each of these fibers has its own characteristic voltage, standard deviation, dwell time, so on and so forth. We capture that information, and then we apply probabilities and say, okay, these 4 5-mers, those are the most likely 5-mers to come after this 5-mer. So we're narrowing the space through a probabilistic model of what 5-mers are going into it. And that's kind of what this is showing here. We have our samples, our events, and our sequence data, following the probabilistic path through the 5-mer options. Okay, enough with the mathematical models here. Read lengths, much more simple stuff here. The cool thing about Oxford Nanopore is it doesn't care how long your DNA is. So as long as you have a properly formed library fragment, it'll sequence it. So if I have a library with fragments on average, in this case 6,000, the reads I'll get out are on average 6,000. This is fairly consistent. A lot of folks have seen this before. So it's pretty cool here. All right. This means that you can even get full-length genome sequences. So lambda DNA can go in. You can get full-length lambda DNA back out, assuming you have delivered properly formed 48,000 base pair fragments to the flow cell. And we've seen that. They're not as common because they're difficult to get to the flow cell, but they're certainly there. Okay, so new things coming for the MinION. Yes, absolutely.
Yes, I just want to ask, when you come around the hairpin and are going back on the other side, I assume that there's some sort of algorithm that knows you are opposite the base from the other.
Actually touch on that probably after the break, on kind of the data structure and what's really in these files and the hairpin structure there as well. Okay, so I've talked about the Mark MinION, told you all about what it does. Here are some new things that are coming out. Fast mode. Currently, we sequence on the Mark 1 flow cell at 70 base pairs a second. In our hands, that's 2 to 3 to 4 maybe gigabases of DNA in a 2-day period. Okay, that's awesome. However, fast mode, in theory, should sequence at 500 base pairs a second. And that goes back to why you need that 1 terabase hard drive on your laptop, because if we move up to 500 base pairs a second, we can see 10 to 20 gigabases of DNA in a 2-day run. That's starting to get pretty interesting there. Now, not yet. Supposedly around Christmas time is the rumor that I'm hearing. All right, the Crumpet chip and 2-pore ASIC. The Crumpet chip is a method by which they're reducing the cost of the flow cells. Currently, the electronics are embedded into the flow cell. That's expensive to make disposable electronics. So the Crumpet chip, in theory, will work with only the fluidics and part of the ASIC in the flow cell, and the rest of the electronics will be actually in the MinION. That reduces the overall cost of the flow cell, which right now runs about $1,000, so it's not that inexpensive. The Crumpet chip should reduce the costs for those flow cells, reduce your disposable costs. The 2-pore system, which is kind of that difficult to see image there, is we talked about all the different proteins they tested. We talked about how graphene pores are inconsistent and they give different signals. But you can intentionally include multiple types of proteins to get different signals to improve your overall resolution. Perhaps some proteins are good at detecting base modifications. Some sequence slowly, some sequence fast. You can group all this data together and get better, potentially, sequencing performance. They also have the Voltrax liquid handler. This is kind of a cool looking thing. I've only seen it once. It is a dedicated fluid handler for Oxford Nanopore. It's about the size of a pack of cards. It's got little wells in it. And you put your different reagents into it, and it's got what looks like a Game Boy screen. And you can actually see little droplets of liquid moving around. They have a current that is somehow manipulating these droplets of liquid, mixing your reagents together, and making magic happen. I have no idea if it works, but it's very cool looking. So nanoliter volumes, electrical field manipulations, and this is going to be for the MinION and the Promethean. Now, what is the Promethean? The Promethean is their new, upcoming, sometime in 2016, early core level sequencing device. This device has 48 independent flow cells in it with a sequencing speed of 500 base pairs a second. Each flow cell should have 3,000 pores, which results in 144,000 pores on a fully loaded instrument. This supports on-site base calling. Right now, the MinION is on cloud-based calling, so you have to have an internet connection to do base calling, which is significant for some of the applications we'll talk about later. And they have this run until mode. So one of the things they've been touting, I personally haven't experienced its use, but they have a system that's supposed to reduce the costs by allowing you to buy time on the sequencer as opposed to reagents. So let's say you want to detect copy number variations in a particular strain of a bacteria. You don't necessarily need a lot of coverage to do that. So you start your run until sequencing, you start getting real-time data, run, until you're confident that you detected what you're looking for, and then you stop. And then you stop paying for reagents, you stop paying for time. Maybe it only takes an hour to get to that data point, 2 hours, 5 hours. Supposedly, this should reduce your costs. This is not available yet, so I can't speak to how well it works, but this is kind of where they're going to, making a very new and different paradigm for how you're going to perform sequencing science. Okay, seriously, does it work? This is the real question everyone wants to know. Okay, AGBT, as seen, flash, you saw the spinning little guys here. Everyone likes to say, oh, this is so awesome, you must have it, but I've been, saying it's so awesome, but I don't know. So here's the answer. Recently, our group has been involved with the Marc Consortium. This is the MinION Analysis and Reference Consortium. The goal of this group is to evaluate and display the functionality or lack thereof of the Oxford Nanopore MinION device. We are also been publishing as part of the consortium in the F1000 research. You're encouraged to, if you do decide to do nanopore sequencing, consider using the nanopore sequencing channel. I apologize, I am selflessly self-promoting here, but I do want to mention this. This is the analysis channel that aims to be a resource for the rapid publication of nanopore data and analysis for benchmarking results in order to drive forward the science of nanopore sequencing. It's an open access, immediate publication journal. It's open source, and it publishes all data, good or bad. Currently on the Nanopore channel, we have 3 articles, 3 posters, and 5 slide sets, and we're quite keen to have more articles on there. The Marc Consortium, back to that topic again, currently have around 100 people signed up for it. There are 20 or so groups that meet on our weekly phone calls. We have 7 experimental groups and several analyses groups, and we host a data hub which has access to many sources of nanopore data. If you want to look at it and not get the nanopore, that's the place you want to go. The goals of the consortium are to provide a consistent data set with tightly defined protocols. From growth of the flow cell loading, this is community driven, multi-lab. Multiple optimizations are being experimented on right now. We want to explore the consequences of changing the protocol and improve data analysis. So if you're an analysis person, you want to play with nanopore data, give me a call. We definitely are interested in speaking to you. Ground rules. Any data you're going to generate must be shared with the consortium and must be openly available. We're going to have open discussion calls. There's a weekly phone call. This 8 p.m. is actually UK time. It's 5 o'clock US time, New York time. You're not constrained to publish your own data, but do be careful that you're sharing this data with the consortium and you don't want to mix or confuse what's consortium relevant and your own data relevant. So. The seminal Marc article, and this goes back to the does it work, is the MinION Analysis and Reference Consortium Phase 1 data release. This is data generated from E. coli from 6 different labs to evaluate how well this flow cell actually performs in the hands of real researchers from around the world. So we're going to kind of go through some of these figures. They're not quite as clear as I was hoping, but we'll touch on some of the major points. Reads. All right. Up, down, up, down, up, down, up, down. The consistency of the flow cells is okay. It's got a lot better than it used to be. Some flow cells generate a lot. This guy here and this guy here, these generated 1.5 and 2 gigabases of data respectively. And then we have this guy here, which generated a few hundred megabases of data. Okay, so you have to be a little bit wary in terms of yield. Reads is the number of reads. These are events. Events are each 5 more going through it. They're obviously very correlated. Read lengths, more consistent. These are all fragmented to 10,000 bases. And while there's some variability where the read lengths are shorter, correlated with lower quality runs, for the most part, if the run runs, it'll have the same length of data. It just might yield as much data. Performance over time. The flow cell dies over time. This is just unfortunately the rule of it. It typically runs for 48 hours for our run here. You'll see this bump going on here. What happens is, in the initial start of the run, the software picks which pore out of those groups of 4 is the best. So it picks 512 good pores. And it starts sequencing from those good pores. At 24 hours, then it switches over to another set of pores they say were the second best pores. And that's why it's this bump. So as those good pores started going, not so good, we change over, we jump up again. There are some custom scripts that exist right now that you can change the time which this pore selection occurs. But those are not currently implemented into native Oxford software. Total yield, unsurprisingly, goes down. This is again the same time point we had the reselection of pores over that 24-hour period, and the number of active pores goes down. For some reason, it doesn't turn on a pore once the old pore died. So that's something I think they're working on, but as a pore dies, they don't replace it. And then that 24 hours is when they actually bring it through there. Yes?
How do they turn a pore on or off?
That's a very good question. I don't have the answer to how they turn it on or off. It's something mechanistic within the flow cell itself. It's not really on or off. Perhaps I'm using the wrong terminology. It's collecting data from it. The pore still does stuff, but you can only collect data from one pore at a time. So I think it's more of a shift of data collection rather than a shift of activity. Does that make a little bit of sense?
I'm thinking that maybe the pores, if pores, are they each independently separated from each other in terms of the voltages they're reading from? So that when a pore is off, what it actually means is that you're just not measuring current from that pore.
You're not measuring current from that pore. Yes, that is true. When the pore is off, you're not measuring current. It still might be taking DNA through it, but you're not collecting data. Now, spatially, exactly how far apart they are, whether there's bleed through or interaction between the pores, I don't know the answer to that. All right, the relationship between pores and yield. Now, one of the things that we do when we first get a flow cell is we go, gee, let's see, is this flow cell going to be a good one? So we look at the number of pores that are present initially. So here we look at the initial G1 pores, that's the best pores. The higher there are, okay, 100% is 512, 80% is less than 512. If it's good, but there is maybe a little bit of a trend to more pores over 450 to better yield. Seeing a little bit more clearly over there the relationship between events and reads. And of course, reads and events are correlated as well, fairly well. That's not a big surprise. So the number of reads generated and the number of events generated for this data set are very close to each other. Okay, now 1D and 2D base calls. I mentioned there was a forward direction and reverse direction, template and complement sequencing. 1D reads are going to be just the template. 2D reads are your consensus reads. These are reads that read through both the template and the complement, and then were aligned by the software within the Oxford Nanopore into a consensus read, and we'll touch on why that's important momentarily. So the longer the 2D reads, the longer events. Not a surprise. These are totally related information. The number of cameras are in there affects how long the 2D is. The actual read length. Template complements tend to be a little bit shorter. That's the tail end of the sequencing run. So you have more frequently the pore stops sequencing or the protein falls off or the DNA comes out or whatever happens, negatively impacted the length. Your best reads are going to be your 2D passes that perform the best. They go through the whole DNA. As a consequence, they're larger fragments of DNA. Number of bases sequenced, there's the fewest number of bases in 2D pass. It's the filtering function going on there. We have a Fred-based filter. Anything that doesn't pass a Fred score will be removed. There's a lot of bases still that go through just template, so that's why that's so many more bases there. Now base quality. That goes back to that 2D, 1D thing. Look how much lower the base quality is for those template and complement reads. And then how much better it is for those 2D reads. That's because Oxford's a fairly error-prone system, as much as 25-30% error at times, which is not good. But by building a consensus between both the template and complement, we can really significantly boost that error profile into something maybe more usable. It's still quite high, though. Base quality decreases over time. This is relating to the failure of the flow cells. They also do worse. They don't just shut down. They also perform worse. They are worse at actually pulling those events out of the data. Errors. Okay. This is the real meat and potatoes, I guess, that people are really interested in for the Oxford Nanopore. So percentage of errors. We have fail and pass reads. So we'll just look at the pass reads, really. Those are the most interesting ones. And we have the total, the mismatches, the insertions and deletions. The percentage of error total is, even for passing, 10% or so. So that's a 90% success call, 10% are failed. That's fairly high. Certain algorithms, so that's these other points here, this is expectation maximization, we'll touch on that after the break, can really improve the performance. However, there is a sacrifice, so you actually see fewer missed calls, but a slight increase in insertions and deletions when you use this expectation maximization function, but it does, to some extent, really improve the quality of the base calls to do that step. Other important points on this slide, not so much, but suffice it to say there's a fair number of errors. Truthfully, insertions and deletions are what dominate the errors more or less. And there are ways of improving it, but they're still going to be quite high. So 10% is a reasonable expectation for the quality. You do get reads with proper algorithms down to 5% error rate, but you're not going to hit that 99% accuracy that you'll see on Illumina instruments.
And when you say 5%, you mean 5% of all bases?
Are wrong.
Bases called 5% are wrong. It's
Yes.
not reads or anything.
Yes, it's bases, yeah. Now, there's some stuff on reads. We'll get to that later. All right. Okay, GC content. This is not all that interesting. Best perfect subsequences, these are the length of the reads that have no errors in them. So on average, your best perfect subsequence of your good reads are going to be about 100 bases. So in typical use, you can expect to have segments of 100 base pairs that have no errors in them on average. Some will be upwards of 300, 200, and there's still a fair that are much less than 100. So that's the length of span you can expect to be perfect. Okay. It is 10 o'clock right now. Okay, so we're going to go through a couple case studies. You've seen what it does, what the Marc analysis says about how it performs. Let's see what some people in the world out there are actually using the instrument for. All right, outbreak monitoring. This is a study done in the UK, monitoring with both Oxford Nanopore and MySeq an outbreak of salmonella in a hospital setting. They noticed an increase in patients, and they started to collect samples off both the patients with the outbreak that was acquired in the hospital and other members from both the hospital and the surrounding community. Over the course of several weeks, we had a large number of cases. Ultimately, about 29 cases were acquired in the hospital over the course of several weeks. So this is 612 all the way to 7.4. Each red node represents part of the core outbreak. These blue nodes represent non-outbreak people that were either possibly in the hospital or derived from the community. And then we have this mini outbreak, if you want to call it, that was in the nursery at the hospital as well. They are related by their SNPs, and you can't see them, unfortunately, it's too pale, but there's actually values on the lines connecting the nodes here. That's the number of SNPs that were differentiated between the different isolates. So these guys are all the same. These guys all have a number of SNPs that are different from 1 to many tens, over 100 different SNPs. This data was generated on MySeq. Okay, I don't want to say this was done from Oxford, but this data was generated from MySeq. That's why the SNP resolution is so good. Okay, here's why the Oxford Nanopore is interesting, however. Identification of a salmonella strain in less than 12 hours, excluding culture time. Okay, they want to be very clear about that. But once you have a culture from an infected individual, 12 hours from library preparation to sequencing to identify accurately the strain of the organism in it. The first reads are mapping to salmonella within 30 minutes of the start of run. As the run continued, more and more reads mapped to the salmonella strain, both from the outbreak and non-outbreak patients. So it's a very rapid, potentially useful for point-of-care monitoring of outbreaks. They used phylogenetic placement to identify the strain. Now the reason why this was useful is because the high error rate in the Oxford Nanopore, you can't necessarily go, I know based on these SNPs exactly which one it is. But as you accumulate reads, you accumulate data over time, you're going to converge on a SNP pattern. More and more reads will match more closely the potential strains. So over about an hour's long period of time, they were able to narrow down on this phylogenetic tree Salmonella, Enterica, Cerevar, interditus as their outbreak strain. That's the red. The non-outbreak strain is actually distinctive. These are confidence bars, no confidence, no confidence. Now we start seeing a much more confident selection of those particular strains for both outbreak and non-outbreak. All right, next. I don't have a paper for this one, but there is some YouTube videos you can certainly watch if you are interested. Point-of-care sequencing. It is hard to get Ebola out of Africa, and that's probably a good thing. So how do you do genetic sequencing on Ebola if you have a hard time shipping Ebola samples out of Africa? This work was done during the 2014 and the 2015 outbreak of Ebola in Sierra Leone, Liberia, and Guinea. And this work, what it did was it created a terrible slide. Okay, it created a opportunity to bring the lab to the Ebola. One of the researchers on this group was jokingly saying their lab was almost as good as the Broad. But what they did was they packed up thermocyclers, qubits, different measurements for monitoring DNA, all of your reagents in a box, and they were able to carry it with them onto the airplane in a bag. So that's pretty significant. You can't really pack up a HiSeq, maybe a MiSeq, and carry it with you. But the size of this instrument really allows you to bring it into locations that you can't necessarily bring full-fledged sequencing into. And so the researcher in this group actually ended up leaving all this stuff there available for the researchers in the location to continue this work on monitoring the Ebola system. Now, they said they didn't have that great of a time. This is why it's almost as good as the Broad, right? Because we have all these horrible insects they were complaining about. It was the very interesting experience for the researchers, and I encourage y'all, if you get a chance, to go listen to the YouTube video of the experience of these researchers and sequencing DNA in Africa. They mentioned a time where they were doing a prep and a mosquito actually landed directly into the tube, which is not a good thing. All right, so what they did was they actually took the Ebola samples, they did a tiled PCR, RT-PCR amplicon approach, looking at different fragments across the Ebola genome, amplified it up. Now they did validate with MiSeq, they weren't relying only on Oxford data because it was still in testing phases. They had tried different panels of amplicons. This was a standard amplicon panel used for standard Ebola identification pipelines. They tried 11 reactions, different sets of things. They ultimately decided that 11 different markers was the appropriate style for the sequencing. And they got a fair amount of coverage across the genome, even with all these markers. They only got about 89.1% coverage using the full marker panel, but they were actually able to cover the genome with larger amplicons with about 98.4% of genome coverage. They had very high total coverage of the genome, over 100 in every case. That's quite good. That really facilitates base calling other pieces of information, because the more pieces of data you have, the better you can resolve high error data. With their study, and they used a couple of algorithms to help get these numbers down, their mismatch rate was about 10%, which is pretty typical for the Oxford Nanopore, better than some, worse than some. This is their protocol, starting with RT-PCR. This is the actual real time. So assuming you have the patient RNA samples, so we're going to exclude how long it takes to get that. 3 hours for RT-PCR, an hour for quantification and pooling. Library preparation, 2 hours. The sequencing, they ran from 15 to 60 minutes. Uploading the data. This was actually not trivial for them. There's not very good internet in Africa. So they were using portable Wi-Fi adapters to upload the data, and it actually turned out that the Wi-Fi uploads are more expensive than the Oxford Nanopore sequencing. Base calling, 20 minutes, and then this pipeline with the marginal line, which we'll discuss towards the end of today, went through, called variants, did inspection, created consensus, and they were able to create a tree and map which strains of Ebola were present. Now, the interesting thing that they found was this strain here. These are all sequenced by the MinION. These are all sequenced by the MinION. Previous work from other researchers had thought this strain, which was present in Sierra Leone, had died off, but the Oxford Nanopore researchers actually found it in Guinea. So they were able to show that this strain was actually moving around more effectively than was previously thought. All right, next case study. Isoform analysis. So with the long read sequencing, you're able to get structural elements that you can't get on short read sequencing. So if you want to know how the actual structure of isoforms in an RNA transcript are going to be, and you want to know the connectivity, long read sequencing is a really very good approach to do, which is quite established now on the PacBio platform, but it also can be done on the Oxford Nanopore platform. One of the nice things about isoform sequencing is the error rate is not as negative to it as with SNP calling. We're trying to organize large pieces of DNA rather than calling individual bases. So what this group did was it looked at 4 highly variable Drosophila genes. I'm not a Drosophila person. I don't know what these genes do. I know that DSCAM is very important, and that's actually what we're going to cover here. So, but the important thing to note is that there's a lot of different exons that can be included in the final transcript. This is a very good choice to look at, very good way to understand connectivity, because there's many different isoforms that can be generated from these different gene sets. So, unsurprisingly, many isoforms can be detected by the MinION. So, this is just the DSCAM. This is not all of the different genes that were used. They were able to generate more than 159,000 reads, which is quite a good number. It's not going to allow for transcript counting like you would expect to see on a MinION Illumina device, but it does allow for isoform detection. There were about 78,000 templates, 48,000 complement reads, and 33,000 2D reads, of which 28,000 were mappable. So we really narrowed that usable data space down to about 20,000 reads, which is disappointing, but not unusual for this instrument. They were able to detect more than 7,000 different isoforms, but they weren't able to achieve read depth saturation. So I'm not going to go through all of these images quite in depth, but basically what this is here, this is a study, say, this is the complexity of each of the transcriptome they discovered. They did not discover very many isoforms, only 1,000 reads were sampled. They discovered more isoforms and 15,000 reads were sampled. This is the expected complexity, this complexity here. So they're really expecting to find closer to 5,000 different isoforms when a smaller number of spaces were sampled. So that's what they mean by sequence saturation isn't reached. That's why counting isn't possible on the MinION device. If you were to do this on MiSeq or HiSeq, you would see this value and these values would be much more convergent. They found there was a significant difference in coverage of all of the different possible exons present in the DSCAM gene. In fact, this exon right here, they didn't even find it. They range from less than 100 reads per exon to more than 2,000 reads per exon. And that may be biologically relevant. There's very likely that certain exons are selected more often in these transcripts than other exons. What they found was that there was, for at least some of the exons, they matched the MiSeq's results. So if you do the same project, looking at MiSeq sequencing, getting much greater depth of coverage, much greater information about how many exons are present, we find that the frequency of exons 4, 6, and 9 from the DSCAM gene are approximately the same. You can't really see 4 here as the frequency in the MinION. So at least for some things with high coverage, we can see consistency in the biological relevance of the data. All right, genome assembly. Genome assembly is very important. If you have an organism that you want to know lots of information about structure variation, or you want to know how the pieces fit together, or you want to know other things that require a whole genome, you're going to need to assemble it. However, assembly is not a trivial problem. When you use short-read sequencing, like Illumina sequencing, what you have is you have all these little tiny pieces that map, and you have some pieces that are fusions between 2 different regions. Now, if these R's represent repeat sequences, how do I know that this R piece goes here and not over here? Short read sequencing doesn't give us that information. It's very difficult, and we have to have very, very high coverage for short read sequencing to get resolution across highly structurally interesting areas at an effective level. This means that we're not confident. How do we go through this order? Is it A to R and then to B? Is it R to C? We don't know. We can't resolve it. So we have a map that's not accurate. Long read sequencing, however, as generated by PacBio or Oxford Nanopore, can span many of these complicated regions, giving us confidence of the connectivity of those different regions, allowing us to create a map with consistent path. Ultimately, we now know that A to R to B to R to C to R, and you can read about this in The Advantages of Smart Sequencing, which is about PacBio. All right. So from that study that I just brought up, which was looking at a yeast genome assembled and corrected as well from Oxford Nanopore data, we can see that the Oxford Nanopore significantly outperforms Illumina data. So if we look at the perfect assembly, this is because this yeast strain has been sequenced a million times, and we are very confident on its sequence. We can see the contig length in N50 is much lower for Illumina. That means we're not able to put it together in as many solid pieces. We have more pieces that we're not quite sure where they go together. With the Oxford Nanopore, however, we see much larger contigs and much greater contiguity of the genome. So we're able to put that genome together into fewer pieces. It's less of a jigsaw puzzle. All right, structural variant detection. This relates back to the assembly. To detect big structures, you have to have big reads. So structural variants in cancer. Now these actually looked at smaller fragments. These are 1.5 thousand base pair amplicons. That's smaller than what you would hope to see with large fragment DNA sequencing, but larger than what you're going to have small DNA sequencing. These are short reads that can be sequenced too, and what we see is that they selected regions of a genome where there were known structural variations. This is a pilot study. And they used 11 structural variations in one control sequence. For sequencing the smaller reads, 1.5 thousand bases. The quality of the bases is quite similar to longer reads. There's no difference in performance using the shorter reads. And some of those samples had about 500 reads for it, which is quite low for what we'd hope to see, but still usable. And what they were able to find is, unsurprising, they did validate. They were able to find translocations within the genome. Okay, it's very difficult to see here. Where we have a movement, where we have reads that maps. So if you look at these bases here, they actually map to that region there. They're able to find deletions. That's kind of deletion right there, where we see large segments of DNA gone, as well as inversions. So even with a low number of reads generated, you can still detect these structural variants fairly effectively with these smaller fragments. All right, microbiome profiles. These are all papers that are either in press or imminently in press. So please, if you're interested in any of these manuscripts, I will certainly share them, their location with you. Microbiome profiling, this is looking at the 16S rRNA of a synthetic pool of organisms. So they actually pooled together all these guys to create a synthetic population, and they did sequencing. Again, these sequencing reads are actually quite low, which is not going to be all that surprising considering the large number of individual sequences all mixed together. Some of these map better than others. The lengths are somewhat shorter, about 1,000 base pairs long, but that's okay. What they found from their mock community is that they were fairly consistent in terms of the population expected distribution. So what this is all the individuals in the community. They were all mixed together in equal proportions of the rRNA, and some were very proportionally expected where they're supposed to be. Some were not so much. So this is actually 2 times less abundant than would be expected from the population as a whole, or this one's here is about 0.8 times more abundant than would be expected. And that's really not all that surprising considering that Oxford has some problems with GC content and homopolymer runs. The higher the GC content, the harder it is to sequence it. So as we saw the percent of GC increases, the deviation actually goes down more so to -1 here. At higher GC, we see deviation closer to 1. So the average here that performs best is your GC content, about 56%. As the GC content goes up, the mismatch rate goes up. It's not huge. It goes from 0.22 to 0.27, but there is definitely a correlation there. They did not find, I know you see this slope here, that there is a relationship. It didn't pass statistical significance, so they say indels are not significantly affected by GC rate. Maybe they are a little bit, but statistically that's not accurate. And they were able to, they saw that the GC observed doesn't totally match the GC of reference. So what they saw and what there should have been wasn't always the same, and that's because of difficulties with sequencing the GC. But needless to say, yes?
Yeah, I'm just curious, did you also repeat that analysis of deviation compared to expected given typical Illumina-based?
So this isn't my own work. I do not believe they included that in their study. My work, which is on yeast, I have done that, and I have definitely seen there is the higher the GC content, the more deviated it actually is based on Illumina as well. So Illumina does not suffer from the GC bias that Oxford Nanopore does. Also homopolymers, poly A and poly T, also tend to drop out in terms of abundance. And that's because it doesn't like homopolymers based on the nature of the way the DNA translocates through the pore. So.
I think on one hand, you're sequencing a longer stretch than the Illumina. Usually you pick 1 or 2 regions of that 16S, and part of the problem with identifying organisms with Illumina platforms is that it's such a short segment, you may not have enough information to match it against the databases. So I think that problem is solved by this, but I hadn't seen a similar graph looking at that type of, what is sequence versus what is expected. So I was just curious to know how it stacks up.
Sure, yeah, I don't know that they did that analysis, but I mean, it certainly sounds like a worthwhile thing to pursue for this. This guy is in preprint right now, or it's BioRVX servers where it's available, so it's possible that it's something that will be included into later revisions of this manuscript. Okay, so we're going to now watch a video. Woohoo, I can stop talking. Where's my speaker on this thing?
sequencing technology evolved very much during the last 20 years or so. But for many years, we have been forced to sequence only model organisms or organisms that were very much interesting for the agriculture, and of course the Homo sapiens. Nowadays, the technology allows us to jump in the future, and also as a scientist, we can go outside of the laboratory in a place like this and concentrate not just on model organisms, but also on wild species. And this is changing dramatically the way we do study genetics. Genetics is now something that is available for every living organism in a place fantastic like this one.
We are currently in the midst of a biodiversity crisis, and most of the biodiversity of the world is concentrated in the tropical belt. Until now, it was impossible or very difficult to take organisms from tropical areas and bring them to labs in the most developed parts of the world. Now it's possible to take the lab to places like this, where most of biodiversity actually is present, and to sequence them in order to get extremely important information for their conservation. In this beautiful forest.
But it's also the beginning of a fantastic new scientific adventure.
All right, so that was Massimo Deladon and his work on biodiversity. So like with the problem with getting Ebola out of Africa, it's also very difficult to get nanofrogs and other unusual, strange, and interesting organisms out of their local areas, which again is a very good thing. But we can bring the sequencers to them, and as you can see, they were sequencing in the middle of the jungle, which was quite unusual, and I don't think anyone's ever been able to accomplish that. Significantly before, I believe there was a sequencing at sea that Ion Torrent did, but I'm not sure about sequencing in the jungle. All right, and I'm going to show one more little short video in the interest of time. Sequencing in space. So yes, they're sending the Mid-Ion to the International Space Station. They want to use it for monitoring sequence evolution in plants in the space station and health of food on there, so on and so forth. That being said, it's not necessarily as easy as that sounds. So we're just going to watch this one little video, if it will come up right. There we go. Why is it difficult to sequence in space? So this is actually the bonnet comet or whatever. Oh, goodness. That's why it's difficult to sequence in space. We have tubes and tips flying everywhere. Well, they're in an airplane, so. Okay, all right, so we're going to take about a 20-minute break while I get the next section of stuff set up. You know, if you have any questions, don't hesitate to ask, but I will be kind of setting stuff up over there, so you'll have to kind of work with me while I'm pipetting stuff, so.